My name is Dan McMann, and my academic research question is, how can genetically modified trees be implemented in future economies? First, to define terms used in this presentation. Genetically modified describes any organism whose genes have changed due to deliberate human action for a purpose. Genetically engineered describes an organism with genetic material specifically altered by modern technology. Worldwide, humans have shown their ability to drastically change the natural environment. Official NASA and NOAA statistics show that carbon dioxide, a balancing component of Earth's atmosphere, has tipped the scales, reaching nearly twice its historical peak concentration, now climbing from 418 parts per million. One consistent carbon absorber, or sink, is trees. Trees take carbon dioxide from the atmosphere, sequestering it in a solid state as biomass, most importantly, wood. Timber, the unrefined body of wood, serves both human civilization and natural environments. However, recent developments make trees more useful for all inhabitants of Earth. Genetically modified trees have the capacity to help both industry and ecology by creating faster, higher quality products and by stemming the overflow of atmospheric carbon dioxide, along with other benefits. Today, two overarching methods of genetic modification exist: selective breeding and gene insertion. Selective breeding has existed as long as farming and works on the basis that if only organisms with favorable traits, as chosen by humans, produce offspring, the future population will display these favorable traits. As described by T.J. Mullen, senior researcher at Skogforsk, the Swedish Forestry Research Institute, gene insertion is the deliberate editing of genetic material by discovering which genes code for certain traits and tweaking them to be more favorable. This begins with the sequencing of the tree's entire genome and ends with the designed genes being inserted via molecular cartridge or agrobacterium into young sapling cells. Amazing traits can come from the modification of tree DNA. Professor Emily Boyd, researcher at Lund University, studied the use of Empress splendor in Canadian forestry. This particular GM tree goes to a mature size within 5 to 10 years and absorbs 10 times the amount of carbon dioxide per year as a wild tree. An average industry tree takes 25 to 30 years to reach maturity. Additionally, timbers from GM trees can be specialized to certain sectors within the industry. Timbers have been engineered to eliminate knots, increasing compressive strength for use in the lumber industry. Additionally, trees have been modified to produce higher volumes of fatwood, an energy-dense biomaterial, making them ideal for wood pellet electricity production. These foreign genes, however, spark controversy for many. The problem with versatile, highly capable trees is that they can destroy natural ecosystems. Spreading engineered genes into wild populations can disrupt natural cycles and cause the endangerment of many species. This can happen through a variety of means. For example, pest-resistant genes can eliminate wild pollinators, or fast-growing hybrids between genetically engineered trees and wild trees outcompete other phototropic life. With a monoculture of trees, the many working parts of an ecosystem fall apart, leading to collapse and extinction of many species. Lead researcher of biotechnology at Oregon State University, Dr. Stephen Strauss recently conducted an experiment in response to this descent. From 2011 to 2018, his team set out to stably sterilize genetically engineered trees. To do this, they used gene insertion techniques to alter the different parts of flowers. For example, a tree may grow petals where a pollen-producing organ normally exists. Additionally, the time of year flowers were grown has changed. For example, trees could only flower upon stimulus from the late summer, reducing the possibility of pollen spreading to wild trees. Through many combinations like this, 3,300 trees over 7 years of testing produced a 100% sterility rate. The novel introduction of sterilizing techniques drastically increases the viability of genetically engineered trees in industry. Using figures from forestry and timber sector sources, one can create a comparison of unmodified, average trees and safely sterilized trees with characteristics akin to the Empress splendor. As you can see, the genetically modified trees can be harvested more frequently and produce higher quality timber, creating higher revenues from previously barren land. However, the defining factor of a timber company based on genetically modified trees is price. Unfortunately, this variable is unknown. Attempts to contact multiple researchers at Oregon State were made, with no success in retrieving the data. However, as large-scale production of these safe trees begins, price will inevitably fall. Genetically modified trees bring ecological benefits besides the ability to absorb carbon dioxide at an increased rate. Operations such as the American Chestnut Foundation have dedicated decades of research and testing on the resurrection of the American chestnut tree, a once prominent tree in eastern U.S. forests. Through genetic modification of all methods, the organization has created a chestnut tree resistant to the Chinese blight brought by humans before the near extinction of the American chestnut in the early 20th century. To increase my understanding of plant genetics, I undertook an experiment in the Liggett greenhouse. In this experiment, I used a fast-growing radish variant to grow a large sample size of plants as a parent generation. These plants were divided into two groups based on the traits they expressed: a purple-colored stem with small formations resembling hairs on the stem, and a non-purple variant with no hair. Upon flowering, the pollen was deliberately spread across the two sets, creating seeds with parents from both sides. To track the transferring and expression of the genes, these seeds would have ideally been grown and observed for the stem and hair qualities of the parent generation. However, due to circumstances surrounding COVID-19, a backup set of seeds was used instead, grown in my home. The seeds produced 95% non-purple stems and were 100% hairless. This is because the backup seeds were chosen to show the dominance of non-purple and hairless genes. However, the appearance of the purple stem shows how the probability works within plant genetics, and that even improbable events can occur. The seeds from the parent generation would likely have produced similar results, displaying the genetic principles of dominant and recessive genes at work. Many thanks to Ms. Elizabeth Than in the research and experimentation processes.